as Annika said, I'm a professor at Cal Poly now. I was an undergraduate at Cal Poly as well. I love the community. I left for 6 or so years and was at UC Santa Cruz where I got my PhD there. And now I feel like I've won the lottery in that I get to come back to San Luis Obispo and teach young people about botany. So what I do normally in the spring is I am not on Zoom and I'm taking young people out into natural communities all over California and teaching them about the plants, something that Dave Keel has done for the last 40 years at Cal Poly. And it's incredibly wonderful and fun to do that. And I'm one of a great community of botanists at Cal Poly. I'll say botany is alive and well and thriving at Cal Poly. We have Dr. Nishi Rajakaruna, who studies serpentine lichens and serpentine plants and weird soils. And Dr. Dina Grossenbacher is an evolutionary biologist who studies the alpine flora. Dr. Matt Ritter, who studies the San Luis Obispo flora and is a tree guru, knows more about trees than just about anybody else. And then of course, Dr. Dave Keel. And I did want to put a plug out that Dr. Keel's, he is writing an update to the flora of San Luis Obispo, last published by Robert Hoover. And that flora should be coming out next spring. And so we're really looking forward to having a party to launch that flora. And all of us are very passionate at Cal Poly about getting young people into nature and actually looking at plants. And that's what we do when we're not Zooming. So today's talk, I want to talk about natural history in the digital age and how it's more important than ever to be collecting plants and looking at plants and putting those plants into the herbarium. And I'm going to give everybody 2 ways in which you can get involved and help. So I'll first start by putting my talk in the context of what is happening globally, but really in the United States. So about 10 years ago, the National Science Foundation decided that it was very important to fund the digitization of museum records. And they created a coordinating center at the national level called iDigBio, and that stands for Digitizing Bio Collections. And the goal of this coordinating center was to work with all of the different natural history collections in the country and get all of their data online. They estimated that there's between 500 million to potentially 1 billion biological and paleobiological specimens in the United States, housed in universities and nonprofit museums all throughout the country. And so these records, these are natural history observations that have been collected spanning 200 to 300 years. And what we would like to do is take all of that data that's normally locked up inside a museum that only a few people can visit and put that on the internet so that those data are available for everybody to conduct research on and inform more about our world. So since the creation of iDigBio, they have funded these coordinating networks at institutions throughout the United States. And what an institution does is they say, we need to digitize a certain set of records for this incredible research purpose. And the National Science Foundation will give them money to digitize those records and make them available online. And so now you can see that pretty much nationally, we've done a great job over the last 10 years of getting partners involved. And what happens is, as each of these institutions puts their data online, we can aggregate that data. And that's what iDigBio has done. So there is one central sort of data portal that all natural history museum records can be searched in. And so currently there's 121 million records housed in this portal. There's 31 million images of these specimens, and those are coming from 1,600 different institutions around the country. And so this heat map that you see shows the density of those records. And so we really do have a global collection of biodiversity specimens from all over the world. And now we can go to one place and find all of the information about them. So today's talk is about plants and what I do at the Hoover Herbarium at Cal Poly. And in case any of you haven't had the pleasure of being there, this is what it looks like. So an herbarium is just a room full of metal cabinets, and inside those metal cabinets are countless and countless hours of human effort and botanical wonder. So if you open up one of those cabinets, you'll see that it's full of plant specimens that are a dried plant that's been pressed and glued to archival paper. And these records then are filed in a certain way, and they provide evidence of what plants occurred where. And there is a very long and wonderful process that takes place before plants end up in these cabinets. So I'll walk you through that just briefly. So first, plants are collected in the field by a botanist. Those plants are put into a plant press between newspaper and they are dried. Then they come into the herbarium. We take those pressed plants in newspaper and we mount them onto archival paper. And then we go through the effort of taking any information about that specimen that's usually on a label, and we put that label data into a computer so that we can look at it in a standardized way and we can search it in an online database. And after all of that, then we can finally file it away into the cabinets. And so the very first step in this is actually just taking a label, which is sometimes typed on a typewriter or sometimes it's handwritten. The older the label is, the harder it is to read. And we capture all of the information on that label and put it into a centralized database. And another element of digitization is taking a high resolution image of that specimen and making that image available online for everybody to look at. So I'm going to make an argument to you briefly about how this is one of the most fundamentally important things we can do for studying biodiversity on the planet. So I'll make the argument that an herbarium is responsible for just about everything we know about plants, whether you realized it or not. So the herbarium, what can we learn from these dried plants in the cabinet? Well, first I'm going to talk about how we know what we have, and I'll do this in terms of talking about the current diversity that we have, new diversity that we don't even know we have yet, and the genetic potential of these specimens. So just briefly, every single plant specimen that comes into the collection has a label associated with it. And on that label, most typically you have the name of the plant. You have the person who published that plant name. You have your location. So in this case, we're at Black Lake. You often have some kind of description of the habitat, like in wet ground at the edge of the lake. You often have associated species. So in this case, you have juncus and typha and others. And you have the date that that specimen was collected. This one was April 25th of 1947, and it was collected by Robert Hoover, and it was his 7,077th collection. That's a typical label, and everyone is different, and different collectors do things in subtly different ways. But regardless, the two most important pieces of information on this label are the location and the date. And everything else, including even what plant is on this specimen, can be argued about later or determined later. But as long as we have a location and a date, that makes this collection valuable for science. These specimens can tell us almost everything about plants. So we can learn about the plant's morphology. So what are the size and shapes of the leaves? What are the hairs on the surface of the leaf like? What's the flower like? We can tell if it's flowering, if it's fruiting. Many times we can tell color, although color is probably the weakest one as these plants age. We can also tell the size of features. Many times there's actual propagules on these plants, so we can extract seeds from these specimens. And in many cases, people have been able to germinate these seeds. And we can also collect leaf tissue off these specimens and sequence the DNA of these plants. And so what that does is it provides us with a 300-year record of what plants were like at a certain spot at a certain point in time. It's very profound. So the California flora, we have over 5,000 species native to California. And the only way we know that is from looking through collections. In the herbarium, you can look at collections from throughout the state, side by side, and compare them. And so, for example, how would you know if there was two different species of Clintonia in California? You see that there's a white flowered one from the Sierras and a pink flowered one from the Coast Ranges. How would we even know that? You as a botanist would have to travel to all of those places and get there at the right time to find them in flower. And then you could look at them and determine if they were the same species. But these records are already there in the collection for you to look at. And so these sheets, or these herbarium specimens, are really the foundation of how we know anything about the flora of California at all. It's also the case that every single plant name that has ever been named refers to an herbarium specimen. And so there's a whole international society that argues about the rules for naming different plants. And the one rule that has stayed the same forever is that every scientific name has to be based on what's called a type specimen. And the type specimen is the reference for that name. So if anybody ever says, oh no, I think it's this, well, you would have one record that you could go back to and say, if it looks like this, then we can call it that species. And if it doesn't look like that, you're looking at something else entirely. And so these specimens serve as the basis for our understanding of what plant is what, and are our structure for organizing all known scientific names. I'll give you two great local examples of that. So here is a specimen that came into the museum. This is a type specimen of Ceanothus maritimus. And this was collected by Robert Hoover in January of 1948. It was then described as a new species, Ceanothus maritimus, in 1953. He collected it from the ocean bluffs, 1.8 miles north of Arroyo de la Cruz on State Highway 1. And so that's a place that many of you might be familiar with. And if anybody ever wonders, what is Ceanothus maritimus? What is the nature of that species? What does it look like? Do I have it here? They can go look at that type specimen, which in this case is housed at the Hoover Herbarium at Cal Poly. Another wonderful local example was an exciting discovery in 2019. This was the discovery of a new species in the city of San Luis Obispo open space. Some of you might have read about that in the papers. So Chorizanthe aphanantha is the Irish Hill spineflowe. It was just published as a new species in 2019. So the city acquired a new open space property, the Waddell Ranch, and Terra Verde Consulting, did the botanical survey for this. And one of their employees and a previous student of mine, Krista Nelson, found a species that did not match the keys. And when she brought that species into the herbarium and we looked at it closely, it turned out it was in fact a new species, and they named that species New to Science in 2019. And in their publication, when they describe this new name and this new species, they have to designate where the type specimen is. And in this case, you can see the holotype is at OBI, which is the Hoover Herbarium at Cal Poly, and duplicates of it were sent to the UC Berkeley Botanic Garden. And here's what that type specimen looks like in the Hoover Herbarium. So if you ever had any question about that name, here it is on the type specimen. So herbaria specimens tell us a lot about what we have. They're also telling us the things we didn't even know we have. So in 2010, a paper came out which argued that herbaria are the place where new species discoveries are happening. Because what often happens is people go out into nature, they collect plants, they don't really know what they're looking at, or they think it's unusual, but they don't have time to sort it out. So they bring it back, they go through the whole process, they get filed into the cabinets, and then they just sit there. And this paper found that a bunch of new species are sitting on the shelves waiting to be described. Based on their estimates, they projected that 35,000 undescribed plant species have already been filed into the herbaria, and they are just waiting for somebody with enough taxonomic expertise to look at them and say, yeah, that's a new species to science. And based on their estimates, they think that it takes most specimens an average of 50 years of sitting in that collection before an expert can look at it properly and figure out that it's a new species. And hopefully that trend will be reversed in the coming years. Herbarium specimens also store DNA of plants. So what I love about this is that the collectors who collected these plants never imagined that we would be extracting DNA from these specimens when they actually made their collections. And so the value of these records has just been increasing with time. And so now we can use the DNA in these specimens to figure out levels of genetic diversity in populations and how that diversity has changed over time. We can detect evolutionary changes and understand how those changes were made. So what I'm showing you here is a phylogeny of monkey flowers, where it shows the relatedness between each of those flowers. And much of that work came from herbarium specimens that were made in the collection. So herbarium specimens also tell us where plants are. And so I think we often take it for granted where plants are, and because we have books and we can look up maps, but it's a profound and ongoing science. So for example, if somebody said to you, do you know where redwood trees occur? You might say, like, of course, they're on the coast of Northern California. But how would you actually know? And if your life was riding on it, if you had a solid bet going on, how could you be certain about where the redwood occurs? And so did somebody drive around looking? Is there a certain book of on the road trip in search of the redwood? And even if somebody did that, they might say things like there were many conifers with needle-like leaves, or the redwoods were very big in the canyon bottom. These are examples of field notes. And those field notes are unclear. We don't know if who wrote them actually knew what a redwood tree looked like, or if they were accurate in their identification. Perhaps they don't know what a redwood tree looks like, or they can't tell it from a fir tree. So how do we know where the redwood occurs? Well, we know where the redwood occurs because of herbarium specimens. So people drive around the state and they can see, they make collections. And so we can validate where those collections were, that those collections were in fact a redwood. And that allows us then to produce maps like the one you see here with the range of the redwood tree. Here's another familiar plant that many of you might know. This is Calycina obispoensis. And so you can say, where does Calycina obispoensis occur? Is it a rare plant, or is it common? Is it found everywhere, or is it special to San Luis Obispo? Well, here I'm showing you on the red dots on this map everywhere that Calycina obispoensis has been collected. And so this gives you a sense of its range, and then we can distill that range into sort of generalized maps that you might see on many of our online databases like the Jepson eFlora. Another way that we use herbarium specimens is to figure out how plants are moving, range shifts. They also tell us what we've lost when extinctions happen, and also what invasive species are coming into our area and how plants are flowering. So I'll give you a couple examples of those. Here's an example of two species, Camebatia foliolosa and Abies magnifica. And people are interested in knowing, well, given climate change, how are the ranges of these species going to change over time? And so you can take their current distribution and figure out what the climate is like where they currently occur. And then you can project that into the future and say, well, where is a suitable climate for these species in the future? And this is an example of where these two species are actually projected to shift in different directions, where one of them is going to move northward and Abies magnifica is likely to move south. And so what that tells us is that species associations that we're used to seeing might look very different into the future. We can apply that same kind of logic and look at just where we expect species loss to occur. So what I'm showing you here are just different climate range scenarios, and the areas in red are areas where we expect to see loss of species into the future. Here's an example of how we know what we have lost. So marsh sandwort, this is Arenaria paludicola. If you look up the Jepson distribution, it shows a map like this where this species has historically occurred from San Francisco Bay all the way along the coast down to San Diego. But if you look at the specimen records for it, we only have collections of it from these few wetlands dotted along the California coast. One of those areas is right here in our backyard. So what I'm showing you here are collections of Arenaria from Black Lake, which is near the Guadalupe Dunes. And here is a collection of this species from April 25th of 1947. Bob Hoover went back later that same year in July and collected it again. Dave Keel then went in June of 1985 and found the species growing there. And since then, it has not been found growing in Black Lake Canyon. So that's just 3 records, and only 3 records that we have that this plant ever existed in Black Lake Canyon at all. And that's really important for a couple reasons. One, we need to document when we have biodiversity loss. And the second is that anybody who's interested in restoring this species back to its habitat, we need to know, well, where is suitable habitat? Where might it grow in the future? And Black Lake is obviously a good candidate for that, since we know that it occurred there in the past. Another way that we use these specimens is to track invasive species. So many of you might be familiar with Ehrharta calycina. This is perennial Veldt grass. It was first collected in 1940 in San Clemente, and then has just slowly been moving its way up. So it was found in the Napomo Mesa in 1956. It made it to the Santa Lucia Mountains by 1958. And now if you live in Los Osos, you will know that this is one of the most dominant weedy grasses in that whole area. It's completely covering Los Osos. It's taking over the vegetation of Montaña de Oro. Yet, if you look at the herbarium specimens of these plants, we only have 5 records of this plant, which are indicated in the red dot here. And that's a major problem, because we want to understand where this plant is going, what it's capable of. And it's typical that weedy species are under-collected because botanists don't like them as much. But it's incredibly important that we document where this plant is moving. One way that we can even put these herbarium specimens on the map is through georeferencing. And so I'm going to make a plug here for you to help us in our georeferencing efforts. So what georeferencing is it's when we apply a coordinate directly to the specimen. So in many cases, especially with old records, we don't have a GPS point. We have a verbal description of where something is. And now we need to use modern mapping tools to put a coordinate onto that specimen. And so I just searched our database to look for places that don't have georeferences associated with them. And these areas, probably many of you know where these are. For example, 3 miles northeast from the junction with La Ponza Road on Highway 58. Anybody been there? West of the sign indicated west end of Botanic area, highway near Cambria, west of dam site in Lopez Canyon, Pecho Road on the dirt road after the deer sign, Rangers Lookout Station on top of Black Mountain, west of, that should say similar, and then Lone Pond Drive across from San Simeon Creek Road. These are just a few of the hundreds of examples of herbarium specimens in the Cal Poly Hoover Herbarium's database that we don't have georeferences for. And if you live near any of these places, you probably know exactly where they are, or some Google searching will allow you to find those. And so one thing that we've developed, we're calling the 100 Club. The 100 Club is a group of people who are good at geography and want to devote their time to reading labels and then putting pins on a map. And so this network of people is growing. And we now have these monthly Zoom calls where we've got a few locals here like Kristen Nelson and David Krause and Kathy, who all help out georeferencing these specimens. So I'll drop a link in the chat at the end where if you are interested in joining the 100 Club, we'll train you how to use it. And all you need to know how to do is navigate the internet and use Google Maps. Another thing that herbarium specimens tell us is about flowering time. And this is really what I have been most passionate about the last few years. You can look at these records, and just by looking at them, you can tell if the plant is in flower or not. And you know when it was collected and you know where it was collected. And that right there provides us with a historical record of flowering time. And it's really the only way that we can build this historical record of flowering time, to ask the most basic question, which is essentially, is flowering time changing? We don't know if plants are changing. although I'll give you an example that we're beginning to know that. We also don't know what most plants are responding to. Are they responding to temperature cues? Are they responding to rainfall? Is it both? Temperature in spring, temperature in winter? We don't know. It's the most basic biological question for many species, and we don't know the answer to it. We're also interested in, if you think of all of California, what habitats and vegetation types are most sensitive to changing their phenology or their flowering time. And then if a plant changes its flowering time, there are cascading effects on other organisms like pollinators, pathogens, and the pests. And so what we're doing is devising a data set that allows us to address these questions. So I want to share with you one little research example of how we can use herbarium specimens to study flowering time. Here is a wonderful species. This is Streptanthus tortuosus, the mountain jewel flower. It's a gorgeous little mustard relative. It has yellow or purple flowers, depending upon where you are in the Sierras. And Dr. Natalie Love was a PhD student working in Susan Mazur's lab at UC Santa Barbara. And she set out to use herbarium specimens to answer the question about flowering time. What is this plant cueing in on, and how has flowering time been changing? And so here's a way in which we can use these specimens to answer these fundamental questions. What Natalie does was she took the herbarium specimen, she looked at the label, and she turned all the labels into georeferences. So then we could put the point on a map. And then we could take the day that that plant was flowering and turn it into a day of the year. So for example, April 8th is the 98th day of the year. So we could see on a linear spectrum, what day of the year is this plant flowering on? And then the next thing we can do is we can extract climate data from every point that we know where a plant was. And this has become very sophisticated, where we can look at the average temperature and the average precipitation for each month of the year. We can look at years in which there was an extreme temperature from the average. It was either very much hotter than normal or much cooler than normal. And then we can look at the subsequent change in the plants that were collected either that year or the following year. And so doing this allows us to look at climate change in California in very interesting ways. So here is a graph which is going to make the point that warming is not equal across the state. And so what you're seeing on the x-axis there is the year. And so we're looking back in time. Each of these data points is a location where this plant was collected, the year that it was collected, and then what the temperature, the low temperature in winter was in that year. And what you can see is that in areas that are cool, so this is the graph on the left side, there's been about almost a degree Celsius change in the temperature. However, in sites that we considered warmer sites, those sites have increased by over 2 degrees Celsius over the same time period. And so what that tells us is that, okay, different spots of California are warming at different rates. And then we can look at how plants are responding to that flowering time. And so what you're seeing here is, again, the year is along the x-axis and the day that it first flowered is on the y-axis. So any point that is around 100 days, that's going to be a spring flowering plant. That's going to be your March, April plants. And the higher up you go on that axis, that means it's flowering later and later in the year. And so I've plotted these divided into warm region plants and cool region plants. And what we can see is that the red line has a negative slope on it. And what this shows us is that over time, plants from warm areas are advancing their flowering time, or they're flowering earlier by almost 20 days sooner than at the beginning of the century. And for cool regions, we don't see nearly that response. So that one is pretty much flatline. So that tells us that we certainly have seen a shift in flowering time, where for this species, plants are flowering earlier and earlier in the spring. Then what we see is that not all plants are responding the same. And so what Natalie found throughout her work is that plants in warmer regions are actually more sensitive to winter temperature, and plants in cool region are not showing nearly the same response. And so that tells us, that sort of adds this layer of nuance to our understanding about these plants. just because it's the same species doesn't mean it's going to respond in the same way to the same environmental conditions. That actually you have plants that might be adapted to the particular environment they occur in, and then they are capable of responding or not to this new climate scenario. So that's an example of how these specimens have proven to be incredibly valuable for studying flowering time. And as a result of this work and other work like it, I was involved in creating what's called the California Phenology Network, using herbarium specimens to build a historical record of flowering time. And so this is what our goals were. 2 years ago, we were funded in 2018. We're starting year 3 right now. The goal was to look at the whole California flora and build a historical record of flowering time. So our goal was to create high resolution photographs of 1 million specimens, get all of the label data from them, put everything on a map, and then look to see if that sheet was in fact in flower. And this is a collaborative effort. It started with 22 and now we're up to 30 institutions who are all funded to take images of their herbarium specimens. And then all of that data is filtered into a database called the CCH2. This is now our new portal. This is where you can find out all the information about California herbarium specimens. And in this portal, you can search through images and images of California's plant specimens. And so another way, if mapping isn't your thing, but you want to help out anyway, another way that you can help out is that we have a bunch of images in this database that do not have label information associated with them. And so, for example, we need to take this image and we need to translate what's on the label into these standardized fields in our database. And we've tried to make this process as easy as possible. And I'm going to show you one way that as soon as this talk is over, you can just stay on your computer all day long and transcribe herbarium records. So we're using a platform that's called Notes from Nature. And I will also put this link in the chat. The Notes from Nature platform has a bunch of different cool biodiversity or natural history projects that they run. But our project is the one with the California poppy, Notes from Nature, or Capturing California's Flowers. And if you click on the poppy, it will take you to our ongoing expeditions. And so right now we have 3 expeditions going on. One of them is from San Luis Obispo. And so if you want to spend some time looking at records from around the area and transcribing them, I highly recommend this activity. So when you click on that button, you will be taken to a website in which you zoom in on a specimen label, and it asks you a series of tasks. And so there's a nice pop-up windows that educate you about how the specimens work, what they are, and what information we're looking for. There's also a neat little field guide in which we've populated it with all of the weird things. Every label is different. And so we have a frequently asked questions section, which usually answers any challenging records. There's also, if you need any help with a task, it will give you examples of what those characters typically tend to look like on an herbarium label. So you can just do this. It will pop up a record and you type in what it's asking for over and over and over again. And the way that this platform works is that 3 different people will enter the information, and then we create a consensus sequence. And once that's done, we import that right back into our database, and it's immediately available online and searchable in the CCH2. We started this late in 2018, and since then, we've completed 6 of these expeditions. We've had over 10,000 records transcribed by over 1,000 volunteers. It's been really great. And this was particularly great in the time of COVID. So what I'm showing you right here is a graph of the number of transcriptions that we do every day. And right here was the last year in March when the California shelter at home order took effect. And so this just shows you that digitizing records and people being at home on their computer, we saw a huge increase in the number of records. And I think this is really important. Digital data allows people to participate from anywhere, particularly from home. And the fact that we had all these digital resources allowed most of our work to continue largely uninterrupted during this last crazy year that we've all had. So I hope that I have convinced you that herbarium specimens are very important for our understanding of plants, and perhaps I might have inspired you to get involved and contribute your efforts toward this resource, which scientists will use year after year after year. So with that, I'm happy to take any questions. And again, thank you to the Botanic Garden and for all of you for inviting me and spending the day with me. So thank you very much.